Okay, let's get started. So for today's lecture, first we'll review a little bit about DNA replication from the last lecture, and then we're going to go through very quickly PCR, which those of you taking the lab part of the class have already been introduced to. And we'll go over it just a little bit. And then we'll spend most of the lecture talking about cell cycle and cell division. Okay. So again, in terms of starting out with reviewing, we talked again about the fact that the structure of DNA actually lends itself to a simple explanation of how it's able to replicate itself. And that comes from this precise base pairing pattern that you see in the double helix of DNA. And so if we separate the strands and then line up the matching nucleotides again, then you see that we can make two copies from the original double-stranded molecule. And so, and then we went over the experiments of Messelson and Stahl, which showed this pattern of semi-conservative DNA replication. And I've taken the figure and redrawn it to make it a little clearer, I hope. So in Messelson and Stahl's experiment, they just had two forms of nitrogen, a heavy and a light, and they let the DNA go one round and then two rounds of replication. And I pretended that there's three forms here, colored sort of dark blue, light blue, and green. And so this is the pattern of replication that you get in strands that you get. So these two strands come apart, and then a new strand is made that matches, it's complementary, and then those two strands would come apart, and now the green strands are synthesized. Okay? And so this is the semi-conservative mechanism of DNA replication and allows a faithful replication of the DNA molecule. And then we went over the fact that in something with a small genome, like a small circular genome, such as in the bacteria E. coli, you can start with a single origin of replication, so you form sort of a bubble in that double helix, and then you start replicating DNA, and that bubble enlarges until you get two separate copies of the round DNA, double-stranded DNA molecule, each one being identical to the starting molecule. Okay? But then if you have a much larger genome, then in order to get done in time, you start multiple bubbles of replication, and then you get what we call these replication forks that are progressing towards each other, and the DNA is then duplicated again into two double-stranded molecules. Okay? And then in thinking of the exact way in which this replication works, we talked about the difference between a leading strand and a lagging strand. And so this has to do with the fact, a very important principle, that DNA polymerase, which is going to actually polymerize that DNA molecule, can only go in one direction. It goes from the 5' to the 3' direction, and it can't go in the reverse direction. And that forces you on the lagging strand to synthesize it in small pieces instead, and then hook them together. Okay? And so just again, a little bit more detail. The topoisomerase is the enzyme that unwinds the DNA so that the replication machinery can get in there. And then you have a primase that puts down little RNA primers, because polymerase can only start on needs a little section that's double-stranded to get going. Okay? And then there's other molecules that, again, are unwinding the helix and then keeping the single strands apart. And again, actually topoisomerase is cutting the DNA and letting the tension that's building up from unwinding it relax. Okay. So then on the leading strand, then with this little RNA primer in place, then DNA polymerase 3 can get started. And again, it only goes in the 5' to 3' direction. So in the original DNA molecule, this is the 5' end and this is the 3' end. But then on the copy you're making, it goes in the other direction. It's the complementary strand. So it goes the other direction. So this leading strand has no problem, and it can keep extending in the 5' to 3' direction. Okay. But for the other strand, of course, it'd be nice if you could just go this way as well, and both strands could be synthesized together in that same direction. But because polymerase doesn't go in that direction, instead what you do is you generate small fragments. So again, a primer sits down, and now DNA polymerase 3 can come along and then extend that and get to the next primer, and then polymerase 1 removes that RNA, and then ligase seals a little gap. And in that sense, you make it in small pieces. These are called Okazaki fragments. Okay? And then together, those two mechanisms on the leading and lagging strand are able to replicate the molecule. Okay? So some people, of course, ask, well, why don't you just wait for the leading strand to come all the way from the other side? And the problem is then you have this huge area that would be double-stranded on one side and single-stranded on another, and you have problems with stabilizing that single-stranded DNA. You actually don't want to leave the DNA single-stranded too long because it get damaged. So this way, the fork can move and both sides are being replicated, and then you can wind it back up as you do it. Okay, the two new molecules can get condensed again as they go. So then a little bit about genome size, and I think I probably stumbled on this when I was talking in the last lecture because I was trying to avoid the confusion of the word billions, which means two different things in English versus most of the rest of the world. But these are the right numbers here. So these are the size of some representative genomes. So very small genomes, like a small virus, can be just like around 5,000 base pairs, so not very large at all. E. coli bacteria has about 4.6 megabases, 4.6 million base pairs. And then something like a human is 3.2 gigabases, right? But you should notice that you don't want to fall into this trap that as animals get more, organisms seem more and more complex, the genome gets bigger and bigger. Not really the case. So you can have things like salamanders with really enormous genomes, and some plants with very large genomes, and then some animals like pufferfish with quite small genomes. So you get a lot of variation in genome size. Actually, there's a lot of forces that drive changes in genome size. We'll talk about some of those later. And again, we'll talk about gene number, but even gene number is not a very good correlation with our thinking about how complex organisms are, and complex, of course, is a hard thing to define. But anyway, this is the sort of range of magnitude that you get for genome sizes. Okay, and so that also means that you're trying to copy a lot of DNA when you undergo replication. And so DNA polymerase 3 can go about 1,000 base pairs per second. So it's one of the fastest polymerization molecules, enzymes known. And so for a human genome, if you had just one place that you started replication and tried to go through it, of course, it would take like a month to replicate all the DNA. And so even at that speed, so that's why you start with thousands of origins of replication and then start dividing or replicating the DNA that way. The other thing that you want to avoid is errors, right? So you don't want to miscopy the DNA as you go. And so polymerase 3 on its own has an error rate of 10 to the minus 4. Okay, that's how often it would misincorporate something. The wrong nucleotide would get lined up and it would zip that in. But it has a proofreading function, so it's actually a very large enzyme composed of many subunits. And part of those subunits, actually, their function is to actually, as the chain is polymerizing, it actually can tell whether the wrong nucleotide has been incorporated. That's because the shape of the DNA molecule won't be right when the wrong nucleotide is in place. And so that has a proofreading function, and so the actual error rate is about 10 to the minus 7. So it's actually very faithful. Okay, but if you look at that, that still means that there's a few hundred errors every time a cell divides. Okay, and that's inevitable. Fortunately, most of the errors to a particular somatic cell in the body are usually not going to be deleterious to it. And we'll talk later about what really happens if something goes wrong, how it can still be taken care of later. Okay. And then, so again, if something gets misincorporated, it can actually be detected, excised, and then repaired. Okay. So I talked a little bit. Someone asked also about how the polymerase can tell what's the new strand versus the old strand, because of course you don't want to correct the old strand with the new strand, because then you'd be introducing an error. And so, because of the way DNA polymerase is going along, it's actually pulling the DNA through it. Is a more accurate way to think about it. The topology of it allows it to touch the two strands differently, and so it can correct as it goes. But if it goes by the error, okay, in some cases you can still correct. So in the case of bacteria like E. coli, the DNA gets methylated, which means that after time, some of the nucleotides get a methyl group put on them. But it takes a while for that methylation to occur. So that means right after the DNA is replicated, the old strand has methyl groups on it, but the new strand doesn't. So in fact, if an enzyme can come along and correct, it can actually detect which is the methylated strand and the non-methylated strand. Okay? And so which one would you use to do the correction? Which would be the correct sequence you want? The methylated one or the non-methylated one? Which one? So which is the correct one? The methylated one, right? So the methylated one is the original strand, okay? And if a mistake is made in the new strand, the non-methylated one, you want to use the methylated strand as a template to fix the error that you've made. Okay, so you can do that. It's not so clear how this is done in eukaryotes. There's a couple of mechanisms that are thought to exist. One is that there actually are these nicks in the new strand of DNA that ligase is still fixing, and so when it detects those, it knows that that's the newer strand of DNA, okay? There are some mistakes that get made that are pretty obvious, though, that they're not supposed to be there, and a very common one comes from exposure to UV light, okay? So if you're out in the sun a lot, then one of the things that happens is if the radiation can affect the DNA actually in your cell, and it creates dimers between the thymines, between the T residues, okay? And that creates something that's totally wrong. It's not supposed to be there, and this also bulges out from the DNA, and so you actually have enzymes that can come in, detect that mistake, and then actually cut out that little area of DNA, and then again, polymerase can come along and synthesize the replacement strand. And in this case, it's easy to know which is the mistake because it has this thymine dimer, which is the wrong thing that you're trying to get rid of. And this is actually very important. So if you don't have this repair system in your cells, then in fact, you're going to develop melanomas at a very high frequency because you'll get errors in your DNA all the time, and some of those will lead to problems, and then you'll get actually a cancerous situation. So these DNA repair mechanisms are actually very critical to the cell for its survival and for the survival of the organism. Okay, so, and then we finish up talking about the problem that happens at the very end of a linear chromosome, okay? So it's fine with the leading strand. You can go all the way off the end, okay? But on the lagging strand, you get a problem because in the best case scenario, your RNA primer sits at the very end of the molecule and you start to synthesize new DNA. But when that RNA is removed, there's really no way to synthesize this little area of DNA, okay? Because you need a double-stranded piece to get the polymerase started. There is none. So that means that on each cycle of replication, you're going to lose a little bit of the end of the chromosome, and that's what happens. So when you copy that, with each round, you shorten the end of the DNA molecule. So they get shorter and shorter by a little bit. And so usually there's nothing essential right at the end of the chromosomes. No essential genes are there. But over time, those ends do get cut further and further in. And so the chromosomes have these things called telomeres, which are at their tips. There's often, and in different organisms, there's different mechanisms to protect the ends of the DNA. So sometimes there's long repeat structures that can be cut into a little bit on each replication without damage. But then there are other ends, there's enzymes called telomerases, which can add DNA back onto the ends of the chromosome to lengthen them again. And it's thought that one reason that cells senesce, that they only can undergo so many divisions, is that, in fact, you have this problem with the ends of the chromosomes. And so without enough telomerase activity, eventually the chromosomes start to decay because the ends get damaged. Right? But like I said, that's by far not the complete explanation for cell senescence, but probably an important component. Okay, so the other thing to consider is that when you are replicating DNA, it has to be unwound. Okay, so we talked about how often DNA is wound up into tight structures. So not only is it, of course, a double helix, but then there are proteins called histones that the DNA winds around, and then those histones form even more complicated structures that winds up the DNA. You need to do this because DNA, like in a human cell, if you stretch out the DNA, all the DNA would be about 2 meters long, and you're trying to pack it into this tiny nucleus. So you've got to coil up this DNA, but you don't want to. Basically, you have to avoid knotting it all up in a way that you can't undo. Okay, so there's a fairly accurate way to coil this DNA up. But when you replicate it, you've got to actually unwind it. So you've got to pull it out in the area that's being replicated, okay, and then replicate it and then get it wound back up. Okay, so as the replication fork is going across, you're having to unwind the DNA, but after it passes, you need to pack it back up. All right, and so that's going on at the same time. And then in certain cases, you'll take it and wrap it up even tighter and end up with these chromosomes, these very, very compact structures. And we're going to talk about this in a moment because you need to do this when you actually divide the cell. After you've replicated DNA, you've got to get it very compacted like this so that you can separate the two molecules that you've made between the two cells that are going to form. Okay, and so you take that 2 meters of DNA, and you pack it down into these chromosomes, and in you there's about 23 pairs of chromosomes, but each chromosome is only about 3 microns long, so very small. And the idea is that that's from that condensation. Okay, and then this is what the chromosomes would look like in a typical human cell. All right, and we'll discuss later, you actually have 2 pairs for each chromosome. All right? And there's 23 of them, the 23rd one being the XY, the sex-determining system in humans. Okay, and so now as a quick aside, we'll talk about the PCR reaction. So this is a way that you can use DNA polymerase to actually do something quite useful in the laboratory. And again, I think probably most of you are familiar with this. So normally you would replicate entire big strands of DNA and go all along the length of them. But you can actually carry out a reaction so that you just amplify a particular fragment of DNA. Don't worry, I'm going to magnify in on the different panels in just a second. And so this is called the polymerase chain reaction, or PCR. And Kary Mullis is the person most responsible for coming up with this mechanism, or this technique, and did this work in 1985. And he subsequently shared the Nobel Prize for the invention of this process. So the idea is that you can have a big, huge piece of DNA, genomic DNA, let's say part of a chromosome or something like that. And if you want to just make a lot of copies of a little piece of it, the PCR reaction is a perfect way to do this. So you start by making 2 primers. So these are sequences that are complementary to the ends of your the piece that you want to amplify. Okay, so this would be your target sequence here. And what you would do is you would make primers that match on some point flanking the piece that you wish to amplify. Okay, and so you denature the DNA, so you can heat it up, and the 2 strands will come apart. You put on your primers, which will anneal here at these positions. Okay, so you've designed them to be complementary to those sequences. Generally, when you do a PCR reaction, your primers are something between 15 and maybe 25 nucleotides long. That gives the right length to anneal at the right temperature. Okay, and then you would add polymerase, and polymerase can start at that double-stranded position and start extending. Okay, and it would start extending in both directions. And that's a pretty typical reaction. But then what you do is that you subsequently reheat the sample, separate those strands again, and then have primers that come in again, and they go back in the opposite direction. And by doing that, you can then take your target sequence and amplify it over and over again. Okay? Because remember, polymerase can only go 5' to 3', right? So that's always a restriction. Is that you have to, when you design your primers, you have to keep that in mind. There's an untold number of people who have messed up their reactions because they forget which strand is which when they make their primers. Okay? So you have to keep that in mind. All right. So, and the beauty of this is that you can keep doing this over and over again, cycle after cycle. Okay? And then eventually, what will predominate is that target sequence that's flanked by your primers, and then you keep amplifying that. So you have these primers that are again 15 to maybe 25 nucleotides, and the piece in between can be 50 base pairs to a couple kb, depending on the polymerase you're using, and you can amplify that. But the beauty of it is because it's an exponential reaction, you can make an enormous quantity of DNA here. But the problem is, before we go on about exactly how fast it amplifies, the difficulty with this is that you are having to heat the sample and and then get the polymerase to work, right, make these strands, and then heat it again, and then keep going through this, right, increasing the number of molecules that you have. The problem is that every time you heat it, and you heat it to about 98 degrees to get the strands to come apart, you destroy the polymerase molecule. Okay, so you have to heat it and add some more polymerase, and let it extend, heat it again, add some more polymerase, let it extend. And when PCR was first invented, that's actually how it had to be done, is that you actually had beakers that were different temperatures, and you had to add your primers and then add polymerase after each round of heating when you broke apart the strands of DNA. And the invention, or the advent, or the real breakthrough, though, came in, it had actually already been found, but hadn't been applied to this yet, and that was the discovery of bacteria that could live at very high temperatures. So a famous one is Thermus aquaticus, and it was actually discovered at Yellowstone in one of the geysers. And so it's a bacterium that's able to live at very high temperatures. So that means its polymerase can function or can survive actually being boiled. Okay? So that's perfect, because now what you can do is you can carry out this reaction, you can add all the components simultaneously, put it in a tube, and just have a machine that cycles the temperatures. Okay? So what's going to happen is you'll have your original target DNA, you'll heat it to 98, which will have the strands will come apart, then you can cool it down a little bit, which will allow the primers to find the sequences that they match, and then warm it back up to 72, and at that temperature, polymerase from Thermus aquaticus functions quite well, and you'll start extending. Okay? And then you can just heat it again, break these two new strands, the two double-stranded molecules that you have, and you start the process over and over. But the beauty is now that you don't actually have to add anything new each time. You have an excess of primers and plenty of polymerase there, and you just let the machine cycle the temperatures. Okay? And so then you'll get this exponential increase. So you'll start off with 1 molecule, 2, 4, 8, 16, and so on. Okay? And so by the time you get to 30 cycles, okay, you'll have about 1 billion copies of your starting molecule. Okay? But not the whole, not like you have a big genomic piece of DNA, not all of that. It's just the piece between your primers is being amplified. Okay? All right. So let's see. Let's try a question then. Okay? And I'll give you a few minutes to read it first. Okay? So if we start with just a single double-stranded molecule of DNA that's 3,000 base pairs long, okay, we design primers that are about 200 base pairs apart, okay, and then we use PCR to amplify, okay, and each cycle, let's say, takes 3 minutes, and after 60 minutes, okay, how much DNA will we end up with? Okay. Okay, and I'll put that back up and I'll start the clicker. Just give me a second here. Oops, sorry. Yeah. Let me try to, let's see, sorry about this. It's kind of hard on the screen. All right, I'll let you read that for a second still, okay? And then I have to take it off to put the iClicker on there, okay? So decide how you want to answer, and then I'm going to put the iClicker software up. Okay. Okay, I'll give you till about 30 seconds, okay? Yeah, I'm sorry, I can't have them both up at the same time. Sorry, sometimes the technology is a little bit funky. Okay. All right. Okay, so let's see if we can find the answers here. All right, so most people got this right. C is the right answer, okay? So, and that's that you end up with about 1 million copies here of a 200 base pair long molecule. So why is it that you don't end up with 1 million copies of a 3,000 base pair molecule? So the reason you don't get that, remember, is your primers are 200 base pairs apart. And on the initial synthesis, they may go all the way to the ends of the molecule, but then on the subsequent amplifications, the primer isn't going to sit at those far ends. It's only going to amplify that 200 base pair piece over and over again, okay? So you end up with 1 million copies, but only of those 200 base pairs that are between your primer, include your primer, and then in between it, all right? Okay. In reality, it would be a little longer. You have to include the length of your primer in that, okay, depending on how you did your calculation. Okay, so let's try to go back here. Okay, so that was a digression into thinking about how you go about using polymerase to do something useful in the lab. And now we'll talk about what we do once we've replicated the DNA. Then our next thing is really to partition that DNA and divide the cell into two. Okay. So again, we talked, the whole point of this was that we want to replicate the molecules that contain information of the cell, and the main molecule that's carrying that information is the DNA. So now we've seen a mechanism to replicate that DNA. So now we have 2 copies, but now we need to faithfully separate them into 2 cells. And so that's what we're going to talk about for the rest of the lecture, is how you divide that into 2 cells. All right. And so there's a lot of instances where a cell needs to divide into 2. So for example, and we'll go through these in a little bit of detail. So in order for single cell organisms to reproduce, right, they're dividing from 1 cell into 2, 2 cells into 4, and so on. But then, for example, also in a multicellular animal, when an